In the last video, what we explored were some general aspects of chemical probes, where chemical probes are small organic molecules that are used to understand the molecular basis of biological processes or assemble biochemical pathways. We learned some of the strategies for creating chemical probes, where one of the key strategies was to take a bioactive small molecule, for example, a molecule that's been shown to inhibit cancer cell lines, tag that molecule with some sort of fluorescent group or other handles so that we can easily detect where that molecule goes within the cell to pinpoint the molecular target of that compound in order to describe new proteins within cells or new functions of existing proteins within cells in order to understand how biological processes work. In this video, what we are going to do is go into a specific example of the development and application of chemical probes by looking at this paper by Hughes et al. That was published in the Angewandte journal. And you can find this particular research paper by going into Canvas and under modules for the small molecules in biology module. This is one of the readings that is provided there, and I highly recommend that you take a look through this paper before viewing the rest of this video in order to get a grasp of an overview of what's going on in the paper. And then we're going to go into explaining the specifics of some of the figures of the paper and the purpose of the paper and elucidating some of the details here in this video. So we are going to be looking at understanding how it was determined that Ammosamides A and B act to target myosin, where myosin is one of the common motor proteins within a cell. The way that this story developed is that in an earlier publication, Ammosamides A and B, of which Ammosamide B is the structure shown right here, were isolated from a marine bacterium, and following isolation from a marine bacterium, were discovered to exhibit cytotoxicity against cancer cell lines. And by cytotoxicity, what we are saying is that the compound inhibited the growth and division of the cancer cell lines. So they were cytotoxic to cancer cell lines. And this particular ammosamide, shown here, Ammosamide B, which is labeled as compound number 2 in the paper, was found to have, in the earlier publication, an IC50 value of approximately 300 nanomolar. So what does that mean when we say that the compound was cytotoxic, killing the cancer cell lines with an IC50 value of approximately 300 nanomolar, where nanomolar refers to nanomoles of compound per liter. So what we are referring to here when we say the term IC50, IC stands for inhibitory concentration. So the amount of compound, the concentration of compound that is required to, since we're using the term 50 here, required to inhibit 50% of the cells in that sample. So it's the inhibitory concentration of 50% of the cells. So it would cut growth by 50%, for example. And so the way that the IC50 is generally plotted and shown is that we would have X and Y axes here. On the x-axis is the concentration, and in the case of this, we'll plot it in units of nanomolar, since that was the final concentration range of this compound. And on the y-axis will be the percent inhibition of whatever variable we are measuring. So here, we could be measuring the percent inhibition, for example, of cell division. And if we were to plot the relationship between the concentration of compound, where we could start with no compound whatsoever, and go upwards to, say, 600 nanomolar concentration at the high end of this, what we would find is that generally we would start with really low inhibition, keeping in mind that the measured IC50 was at about 300 nanomolar. When we get close to 300, it will take a leap upward to reach 100% inhibition. And in this curve, this is a very typical curve shape that's seen for inhibition of growth of cancer cells or bacterial cells or whatever model system you're looking at, generally the inhibition is relatively low, and then there's this inflection point where rapidly the cell receptors that are responding to this compound become responsive and saturated, bringing it up to 100% inhibition of that molecular target and resulting in 100% inhibition of cell division. And so the point at which there is 50% inhibition is right here midway up this curve, and that would correspond to 300 nanomole if we look down here at our plot here. So we interpret the IC50 plot as we plot out at several concentrations experimentally what the response of the cell growth is to that particular concentration of compound. So if we measured out the amount of inhibition of cell division at all of these data points represented by X's, representing different concentrations of compound all the way through here, determine to what extent those inhibit cell division, and then plot that, we generally end up with a plot like this, which we could then go back, find the plot of best fit here, and track where the 50% mark is there, where at what concentration are 50% of the cell growth inhibited. And that gives you the IC50 value for this particular compound or whatever compound you would be exploring. So the term IC50 is a really common term used both in establishing how potent a chemical probe is at eliciting a biological effect, and also very, very prominently used in the process of drug development and drug discovery. So if you are pursuing a project later on in the semester for your presentation where you're looking at drug discovery, chances are you're going to come across the term IC50, which stands for inhibitory concentration of 50%, or a very similar term, effective concentration 50, which would be EC50 for effective concentration, which is similarly looking at the concentration of compound that is responsible for eliciting 50% of the effect that can be observed for that compound. So in the case of the Ammosamides, the original work on the Ammosamides after the compound was isolated and identified using a battery of 2D and 1D NMR experiments such as COSY and HMBC, HSQC, and then 1D methods such as carbon-13 and proton NMR. What then occurred was that the investigators were interested in how does this molecule actually function to inhibit cancer cell line division? At what protein is being targeted in the cancer cell that is enabling this compound to effectively halt the cell cycle and halt the cancer cells from dividing? And so in order to tackle that question, in order to track where this molecule is going within cells, what was taken was the approach of turning this into a chemical probe to understand where this is going within a cell, and in the process, hopefully better understand the proteins involved in the cell division process. And so in order to accomplish that, what was done is that the Ammosamide molecule, up here at the top, compound 2 was Ammosamide B, was reacted with compound number 3, shown down here at the bottom. This is a coumarin derivative. Coumarin molecule has this sort of motif that we see right here and what I'm circling with the laser pointer. And the important part and thing to keep in mind about structure number 3 is that this particular compound both exhibits fluorescence. So if this functional group is present within a molecule, the molecule will fluoresce. And then the other cool aspect about this molecule is that it is also recognized by antibodies, meaning that it will bind to an antibody, bind to a specific antibody. And that provides an extra way of detecting this particular compound. We can detect it by 2 different methods. One is that it fluoresces, so we could observe where it is going within a cell as a glowing region of the cell. We can also enable it to bind to an antibody to pull this molecule away from others within the cell in order to track what it's interacting with. And so it was envisioned if we could covalently link Ammosamide B to this fluorescent tag that also is capable of binding to an antibody, that could be a really valuable way to enable us to track where ammosamide is going within the cancer cell. And so the approach here, summarized in text for purposes of your notes and things, is that the investigators aim to covalently link the ammosamide molecule to that group seen here that is capable of fluorescing and that fluorescent group is also recognized by an antibody. It binds to a particular antibody. And I'll abbreviate antibody as AB. And a term that you should keep in mind here is that a group that is recognized and binds to an antibody is referred to as an epitope. So I may use the term epitope, and you'll see the term epitope coming up in that literature paper, and that's just referring to a particular molecule or functional group that is able to bind to an antibody. Unfortunately, in the case of the ammosamide, when it was considered how Ammosamide B could be linked to this fluorescent group, it was noted that Ammosamide B has a couple of amine groups here and here are primary amines within that molecule, and those were anticipated to be capable of reacting with the carboxylic acid functional group of our tag here. And thinking about this bond formation that we would anticipate occurring here, we could envision that there would be an amide linkage take place there. As we've learned back in the protein segment, for example, that amine groups join up with carboxylic acid groups to give those peptide bonds, also known as amide linkages. The same would be true here. And in Ammosamide B, there were actually two amines here and here, two primary amines that hypothetically could react with the carboxylic acid group. And it was important and envisioned that the reaction would occur here between the carboxylic acid group and the amine group, because that would leave the fluorescent portion of the tag intact here. This was our fluorescent group right here. So it was important in considering that the group in the molecule that is fluorescent is not going to be disturbed by the chemical reaction. And in fact, the same moiety of the compound was also what's responsible for binding to an antibody. So it was important that in the reaction, this group be left completely intact, so we have that fluorescence and antibody binding taking place. So the link here that was targeted was the link between the carboxylic acid and the primary amine group. And indeed, when this reaction was run, what was found was that this product, referred to as product number 4, resulted. And we can take a closer look at what has occurred here in this reaction. In this reaction, what we see is that the observed link is a link between the primary amine group that originated right here from ammosamide. So this is our whole structure of ammosamide up top here on our product side. And then it's linked to that nitrogen that was originally the primary amine. And now we have that amide linkage coming over to what was the carboxylic acid carbonyl group over here on the reactant side. So the reactant side was our carboxylic acid. It's become the carbonyl of the amide group over here on our product side. And interestingly, in this paper, it was found that this product number 4, was the major organic product of this reaction. There was not a tendency of the reaction of the amine group to the carboxylic acid to occur at this position, at this primary amine. Instead, this primary amine, as we can see in the product, remained unreacted. And the hypothesis for why that was the case was that this amine, the amine that actually reacted, is less sterically hindered than the other one, and that promoted the reaction taking place at this particular location within the molecule to give our amide linkage here. To confirm the structure of this compound, the group applied NMR techniques, much like what we learned a couple of units ago. They applied, primarily as their tools, carbon-13 and proton NMR first, and that allowed, by proton NMR, for the experimenters to observe for this single compound a number of signals that were consistent with the expected structure here. Likewise, in the carbon-13 spectrum, the number of carbon-13 resonances, indicating the number of carbon atoms in this molecule, matched up with the number expected for this compound, and the particular chemical shifts correlated with the expected chemical shifts of the groups that are seen here that are either shielded or deshielded. Then from there, to provide further evidence for the structure of this compound, we might, if proposing what the scientists need to do here to be conclusive about what structure has been generated here, we might propose that they use 2D NMR techniques such as HMBC and COSY, where HMBC looked at long-range correlations between hydrogens and carbons, and COSY looked at long-range correlations between coupled protons. Now, in this molecule, the most important region to determine was successful in the synthesis was that this link had been formed right here. And for the case of this molecule, when it was considered to use COSY or HMBC, those two techniques were not particularly useful in concluding and confirming that this part of the molecule was intact. The reason is that if we look at what expected signals we would see in this region for a COSY or HMBC spectrum, if we think of hypothetically wanting to confirm that this link is in place here, that we form this new amide bond between the carbonyl and the NH, if we envision and we look at what carbons are directly bonded to hydrogens here, and we think about what correlations those would be expected to have by HMBC and HSQC, or COSY, if we look at this CH2 group here and we think about what COSY correlations we would expect that to have, when we come over to the adjacent carbon here, that carbon has no directly bonded hydrogens, so there's no vicinal hydrogens coming this way. Coming this way, there's no vicinal hydrogens, so that we expect to have no COSY correlations to this set of protons here. For HMBCs, the two protons here would show correlations to here, correlations over to here, but all of that was intact in the starting structure. It does not enable us to connect over to here to like the carbon at position 8 right here, because that is going to generally be too far away or bordering on too far away, particularly since we're separating the two parts by this nitrogen heteroatom. And so that prevented the use of HMBC and COSY to confirm that this link had been created. Likewise, if you look over here, there's no hydrogens at this position, no hydrogens at this position, no hydrogens at this position, and so that made it impossible to look from this perspective, from this side, to come up with COSY correlations or HMBCs to stitch together the structure as well. The only proton that we had here is this one that's bonded to NH, and those tend to behave rather unpredictably by HMBC and COSY. We don't generally see a predictable set of HMBC and COSY correlations from these exchangeable protons like this amide proton here. What they were able to see, though, was what we refer to as nuclear Overhauser effect, NOEs, which we did not specifically go into when we were talking about NMR methods, but what a nuclear Overhauser effect shows is what protons are close in space. So it neglects what protons are bonded to one another and instead allows us to explore, by this nuclear Overhauser effect, it allows detection of protons that are within less than 5 angstroms of one another. And so that distance of less than 5 angstroms would correspond to protons that are relatively close in space within this molecule. So nuclear Overhauser effects are what were measured here, 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 and elsewhere in the molecule to enable stitching together of the structure and provide conclusive evidence that the two groups, that is the Ammosamide reactant, shown here, had been covalently linked with our fluorescent epitopic tag, because we were able to see correlations, nuclear Overhauser effects, between the proton here on the amide and the methyl protons over here. Likewise, that proton from the amide also showed correlations, nuclear Overhauser effects, over here to the CH2 hydrogens, enabling the determination that indeed that link had been formed in the chemical synthesis of these molecules. And so with this tagged ammosamide molecule in hand that had the tag that will allow hypothetically both fluorescence and binding to an antibody, the next steps were to experimentally test this. And we are going to go into that in the next video and look at the journey of how the scientists went about taking this molecule and determining its cellular target, using it as a chemical probe.